This is the quick guide to Laboratory Quiz 2. As usual, the information below is certainly not a complete list, but it does give a few areas to concentrate your efforts on. As usual, anything in the Fox manual or the laboratory supplement is fair game. This is not a complete list, as you know. You're responsible for everything that is presented in Fox, the supplement, or any of the class discussions or videos as they relate to the experiments. So we'll go through this guide. The first lab you're responsible for is Lab 7-8. That is Fox 2.6A and C, and Supplement 1, 2, 3, 4 in class discussion. We actually, for this time around, are only doing supplement numbers 1 and 2. Take special note of a couple things. 2.6A talked about the demonstration in Fox about the xylene in water. So remember, we took xylene, which is a non-polar aromatic ring, and mixed it with water, and the two separated pretty quickly. Then we put potassium permanganate, which is a salt. Since it's polar and the water's polar, the salt dissolved in it. We could tell because it turned the water purple. Then we put oil in it, and that dissolved in the xylene because xylene is nonpolar, as is oil, because most aromatic rings are nonpolar. In fact, all aromatic rings are nonpolar. Then we shook it up and it separated out, and then we put in soap, which is amphipathic. It has polar and nonpolar ends, and that emulsified it and kept everything together. We also looked at the osmosis of the water across this dialysate tubing in response to varying levels of sucrose, and we should know how to predict the direction of water movement and the degree of movement, right? So remember that the larger percentage of sugar will pull water towards it, and the bag will get heavier or lighter depending on the movement of the water. You should know how to calculate the percent changes based on the movement of water over time. You should know how to, which is not on here, but know how to calculate the percent change, right? Oh, there it is, percent changes based on the movement. Know how to calculate milliosmoles, right? So for the solutions that we're typically responsible for, sodium chloride, calcium chloride, potassium chloride, and glucose, from grams per deciliter like we did in the laboratory. You should also know how to determine the tonicity of a red blood cell when dropped in solution based on the calculation of milliosmolarity. If it's below 280 and it's human red blood cells, right, it would be hypoosmotic, and the red blood cell would swell, it would gain water. If the difference was big enough, it would eventually burst. If it was 280, right, that would be isotonic. The red blood cell would not change size. And then if it was above 280, it would be hypertonic, and we would pull water out of the red blood cells and they would shrink. You should also know the effect of molecular weight and polarity on hemolysis time, right? So the smaller the molecule is, the faster it will diffuse across the membrane. So the faster hemolysis time it'll have, and the more non-polar a molecule is, it'll also increase the speed in which it moves across the membrane, which means it would have a faster hemolysis time. So that's Lab 7-8. Lab 12, and we say Lab 11 and 12, but you're not really responsible for the setup. You should understand basically what we did in the Lab 11 on how we set up the lab, what we do to the nerve where we shock it once and change the voltage to show spatial summation. You should know what we do for the muscle twitch. It's the same thing as the nerve by shocking it once, and then how we have temporal summation and the various levels of temporal summation that we do with Treppe, wave summation, and tetanus. You're not responsible for Fox 3.1, 5.1, or 5.2. I did not look at those to find any information. A lot of the information we talked about in the videos and the information for the class materials comes from that information. but you don't have to study this particularly. So you should understand the difference between individual action potentials and compound action potentials and how each one works. Individual action potential happens in a neuron, or a compound action potential happens in the nerve, and it's the sum of all the active individual action potentials. Know the spatial summation of the frog nerve, what we did there. Know the terminology associated with the various conditions, such as subthreshold, threshold, submaximal, maximal, and supramaximal, and what type of voltages and responses we would see in those. Know spatial summation in the frog muscle. It's basically the twitch. Understand the different parts of the twitch. And then know, like I said, the different parts of temporal summation, what happens in TREPI, wave summation, and tetanus from an experimental standpoint, and then what are the mechanisms that cause those, right? Remember we said TREPI is an increase in calcium, and that gives an increase in contraction strength. Wave summation is an increase in calcium, and because it doesn't go back down to baseline, the pre-stretch of the series elastic elements. And then tetanus is the same as wave summation, except now we have the tonic muscle. And so in tetanus, the frequency is high enough to recruit that tonic muscle we see in the frog gastrocnemius. So that's lab 12. Lab 13-14 is Biopac lesson 1, EMG, and lesson 14, the biofeedback. The EMG does have a corresponding section in FOX 5.3, but you're not going to have to read it. You are responsible for the information given in the supplement on EMG from the Biopac, and then the biofeedback. It's like, one or two or a couple pages long. So anything in there you should understand. For the EMG, you should understand what we're measuring. You should understand the difference between spatial and temporal summation and how they relate to the EMG. You should know what causes the differences in the EMG. For the biofeedback, you should know what branches of the autonomic nervous system affect what tissues. So if we have a change in sweat rate, what's affecting that? If we have a change in heart rate, what's affecting that? You should know what happens with EDA and how that works as well. So you should be able to identify changes based on different levels of the heart rate or the changes in EDA, what's causing them. So that's 13-14. 15-16 is looking at all the little discussions for the sensory lab. So we did 2 parts, 3.4 of FOX, A, B, C, D. Those are the general sensations. Also, you should know FOX 3.5 A and B and supplement 1 and 2 as well. Those are the visual ones. So what happens with the eye stuff. You're not responsible for FOX 3.5C and also the following tables. So table 3.1, 3.15, 3.19, and then figure 3.2 and 3.21. So those look at like the eyeball structure and some of the things that I think are more anatomical and just you're not responsible for them. By extension, then, if they're not specifically listed, you probably should look at the figures, and that's true of any FOX, that if they're not specifically excluded, and you're supposed to know that section in FOX, then you should pay attention to those as well. Also, the D1, D2, D3, D4 formula in the supplement, I won't put on the quiz either. So things you should really understand is, in the basic terms, what we're doing for each experiment, what's involved, such as what's test, what did we look at, what do we expect, and what are the associated physiological outcomes. So for the general sensations, right, we tested the touch and temperature receptors and what our conclusions were how touch receptors are distributed very widely in the body and have these very dense, small receptive fields, where the temperature receptors don't have that because you don't need to localize temperature usually. The 2-point discrimination threshold and how you need to excite 2 different receptors to feel the 2 different prongs, and how all of that works with, again, receptive field size and density. How temperature receptors adapt. That was the warm and cold hands in the water and then putting them in the room temperature water, and how the one that was cold felt warm and the one that was warm felt cold, and talking about their adaptation rate and the response, how they respond to changes in temperatures, generally speaking. And also the section in referred pain, where we hit the elbow, and some of the components of that. In terms of the visual tests, you should understand the 2 ways we measured visual acuity, even though you don't have to do the calculations for the D1, D2 eye thing. You should understand that test. You should understand the Snellen eye chart then, and how that works. You should understand about what astigmatism is, and you should be able to understand what the near point of accommodation is and how to use that table to figure out someone's visual age. And then assess it based on their chronological age, which is really just how old they are. The last lab, 1718, that's BioPAC Lesson 3. That's the EEG. And again, the only thing you have to know from that was what we talked about in class. We'll get to it in a second. And also the FOX 3.3 you're responsible for, and that's the 2 reflexes, just the 2 reflexes that we did. The knee jerk, with and without the gindrastic, and then the plantar reflex, and how a negative plantar reflex, I guess a one that's not normal, an abnormal one, is associated with the Babinski sign, right? And so again, the EEG has a section in FOX 3.2 you're not responsible for. So the EEG, you don't have to know much about it. We said it just measures the electrical difference between 2 parts in your brain. If there's no electrical activity, right? What happens with the EEG? It's flat. When does that occur? When you're dead. I want you to know the expected data, not so much with eyes open, eyes closed, but the clasping of the hands when you do that gindrastic, right? That should give us the biggest amplitude. For the reflexes, you should understand the general reflex arc. So the receptor, afferent, integrator, efferent, effector, and how that is specifically applied to the knee jerk. You should understand the reflexes in general, and the differences between the 2 we studied. I just noticed this, but it says knee jerk and patellar tendon. Those are the same reflex, right? It should say knee jerk and plantar reflex, right? You should understand how the reflexes are mediated by the Renshaw cells and what happens with the gindrastic maneuver when you try to enhance the reflex. And you should know how the plantar reflex works as well. So, again, just let me be clear, you should compare the knee jerk versus the plantar. So, and in that worksheet, there was a nice comparison about how one's, knee jerk's monosynaptic and the plantar is polysynaptic, and the knee jerk is simple and the plantar is complicated, and the knee jerk is a spinal reflex and the plantar reflex is a cortical reflex, and things like that. You should understand how things work in terms of when you see a normal reflex versus an abnormal reflex and what would cause that, especially as related to different levels of spinal cord injuries. And again, that's, outlined on that worksheet. So if you took a look at that, then you think pretty much understand all those things. So that's a quick rundown of the different things that you might see on Laboratory Quiz 2.